Hello, my name is Olivia, and you're tuning in for part 2 of the two-part series on thymidine kinase 1. In part 1, I talked about how TK1 is a cell proliferation protein with powerful potential as an oncology biomarker for all types of tumors, including both blood and solid tumors. In this part, I will introduce you to the TK210 ELISA kit by AroCell. It's a unique tool to measure total TK1 protein. If you need a refresher on TK1 as a biomarker, watch part 1 first. If you're interested in hearing about this new ELISA kit, continue on with me. It's important to note before we get started that this product is for research use only in the US and Canada and is not intended for diagnostic use. To measure TK1, there's two different types of assays. There's activity assays and total protein assays. The activity assays measure all thymidine kinase in a sample and can include TK1 and TK2. The activity assay can be difficult for some in the fact that it requires radioactivity in many cases or really expensive closed systems. The biggest drawback to the activity assay is that, as we discussed in part 1, TK1 can be found in many multimeric forms in the serum. Much of that is actually inactive protein, so an enzymatic measurement would miss a lot of the TK1 in a serum sample. For the protein-based assays, previous attempts at producing a standard ELISA have also been difficult due to this multimeric complex issue. When they're in these multimers, a lot of the antigenic epitopes are being masked, and it's difficult to get a robust ELISA readout from that. Seeing this gap and need in the commercial market, a company called AroCell in Sweden has developed an ELISA that addresses the problem and fills this gap. AroCell addressed these issues by creating a monoclonal antibody against amino acid 210, which the kit is named after, and establishing a buffer to dissociate the complexes at an early stage in the ELISA protocol. It's resulted in a more robust ELISA that correlates better with the gold standard of enzymatic activity. The benefits of the TK210 ELISA kit really boil down to three main characteristics. One is that it's a simple ELISA protocol that doesn't require any radioactivity like the activity assays. The second is that the monoclonal antibody is specific for human TK1 and can distinguish between human and mouse TK1, which makes it ideal for things like xenograft tumor research. The third is that TK1 is measured in the serum, so it's a non-invasive biomarker and ideal for things like clinical research trials, so that biopsies aren't required. If you're interested in the 210 ELISA kit to make more accurate TK1 measurements, please reach out to us at Diapharma. This kit, in combination with our apoptosis-specific kit that measures caspase-cleaved keratin 18, would make a really interesting mechanistic panel for monitoring cell growth and death in many drug development or clinical research studies. We look forward to hearing from you.